Welcome back, everyone. Next up is Bioxytran Inc. They are on the OTCQB, trading under the ticker symbol BIXT. The company is a clinical stage biotech with two revolutionary platform technologies. They have an oral glycovirology drug that recently completed clinical trials and achieved 100% viral reduction in 7 days, a feat only accomplished by one other drug in the past decade. Their drug is a galectin inhibitor that neutralizes the COVID-19 virus and prevents it from entering cells. They also have another platform drug being developed to treat strokes. Now, due to the molecule's small size, it has the ability to carry oxygen into areas of poor circulation. Now, I did some research, and the JP Morgan conference is kind of like a mecca for big pharma, biotech, investors, and VCs to meet once a year to share their ideas and network. And every year they host it in the heart of San Francisco near Union Square, and many even piggyback off the conference with side events nearby. So this year it looks like over 10,000 people showed up to the JP Morgan conference and the biotech showcase. So it's kind of like a big biotech networking party that invades downtown San Francisco. So it's my understanding that you, Mike, attended the biotech showcase. So maybe you can give investors some insight with respect to what you might have learned at the conference, as well as any key connections you might have made. Tell us, how was it? And welcome, Mike.
Oh, thank you. Pleasure being here on the Emerging Growth again. You know, this conference, it was just absolutely massive. Okay, you had people in meeting rooms, there were hotel lobbies waiting outside hotel entrances. Everyone was networking, passing out business cards, you know, doing what they do. And then we had parties afterward, you know, great food, atmosphere, you know, entertainment. It was just really a super festive type of event. But it was all centered around really networking and making these real connections with people. Now, there was a part of the conference, I attended the biotech showcase part of the conference primarily. And to help people connect, what they had is this dating app almost, right? So you sign up for these one-on-one networking events, but before you sign up, you got to build your profile, just like, you know, a dating app, you know, so you got to make yourself look good to the audience that you're going to, and vice versa. They have to do the same. So then what you do is you sign up, you send messages to the people that you want to meet with, you schedule a time, and if there's a match and they accept you, well, then the program automatically assigns you a booth. And then you get to this networking table, then you sit there and you give your presentation or you just talk. It was this grand ballroom, and there was literally hundreds of networking booths, and you could actually walk by and just watch people. It was pretty amazing. But it's basically speed dating for biotechs. And what they would do is, you'd see a lot of investors paired with biotechs. You would see people called CROs, clinical research organizations. You know, they want to sell their services to other biotechs, you know. So there's a lot of that networking. But my goal at this conference was to basically find some big pharma connections, establish some sort of, we really need to get, we're at the stage where we can do some licensing. So, it's important to make those personal connections right now. But most importantly, all biotechs are always looking for money. So we needed to find those institutional investors that like what we do. So this should give investors a pretty good idea what this conference was about and, basically what we're trying to do. Now you're asking about like the actual connections that were made. Look, I came back with just this massive wad of business cards. I met big pharma players, I met biotechs, I met institutional investors, and we even met the guys that help you find the institutional investors, Basically, there's just a whole myriad of people that we talked to, and I even had the chance and opportunity to sit down with a couple research analysts. Now, I think the fruits of all this labor and all the, because it takes time to follow up, I think it's going to actually happen pretty soon. Now, the other side, remember I said there's the JP Morgan conference, and then there's the biotech showcase. Now, the JP Morgan conference, that security had like high, I mean, that venue had a high amount of security. So when you pass by the outside entrance, there were these guards guarding it, like you couldn't get in. So those were for like the big pharma players and the NASDAQ type companies. Now, we're not NASDAQ yet, hint, hint, stomp, stomp, but we're on our way. And I think by the next JP Morgan conference, we might be able to get into that venue. But for the most part, we stuck to, we didn't make the cut, and I stuck to the biotech showcase. So maybe next year, we'll see what happens. But I'm really excited to take advantage of all these networking opportunities that I got at the conference.
Sounds fun. Speed dating for biotech. Yeah. So what was your biggest takeaway from the conference?
Anna, overall, it was a really good conference. I'd say my biggest takeaway is because, I had attended this conference before, but the biggest takeaway was the amount of money people that were there. Before, money people were chasing deals. But this time, there were a lot more biotechs chasing money, and there wasn't as much money out there. And when I mean money, I mean these institutional type investors. So I guess my biggest takeaway is it's really hard to find money now, and the scales have really tipped. And I look at that as like a challenge and as a way to be just more creative and thinking outside the box. Because what I did notice is there were quite a few biotechs out there, and I don't know if people look, but there's a lot of biotechs trading well below cash, and they have no pipeline. So if you think outside the box, why not? We could build out a pipeline with some of these biotechs would consider chump change. Why wouldn't we want to start talking about these things? So look, the bottom line, we made a lot of connections. We got a lot of great prospects, but the brutal reality thing is that the things are a little bit tighter than they were 3 years ago, the last time I was there.
That makes a lot of sense. We also saw on Twitter that while you were there, you met up with the Nobel Prize winner, Carolyn Bertozzi.
Yes.
That's pretty cool. So I saw that last year she won the prize in chemistry. So how was that meeting her?
Well, before I get into the meeting, let me give you a little backstory on actually how we met. It's really about networking. I saw that she got the Nobel Prize, and then I basically started following her on Twitter. You see, her field of research is glycobiology. What's our field of research? It's glycovirology. You see the cross-pollination there. So what happened is I responded to she tweets. I respond to the tweets. I made some comments, and basically some of my comments caught her eye. And then all of a sudden, I had this Nobel Prize laureate following me. I thought that was kind of cool. So she must have liked what I wrote. So I thought, heck, I'm going to give this a shot. So before the JP Morgan conference started, I reached out to her on Twitter and said, hey, you going to be in town, basically? And she says, yeah, I'm going to be here. You want to come by and meet? And we're like, oh, okay, let's do it. So right before her speaking engagement, which was hosted by Eli Lilly, did a lot of networking there. We broke bread and got to know each other a little bit. My conversation started out with, look, I know what you've been doing in glycobiology. I think it's really cool. And the part that I thought was cool is one of the things that she was really directly responsible for was the imaging of T cells. And when you can image a T cell and see how it acts in real time, you have a better understanding of what they're doing. And the better understanding of what they're doing was in cancer. And that is basically her specialty and what drives her. And the truth is, we're doing all this stuff in coronavirus and viruses, but that's my specialty. That's something that I'm very passionate about. So, we bonded over that. We talked about something called the galectin effect. I think I've talked to you about that. But the galectin effect, there's overlap between cancer and virology. And the effect in cancer that galectins have in cancer and the effect in virology, it's very similar. So we discussed that, and that was pretty good. But, here I am. I'm talking to her. I have her attention. Of course, what am I going to do? I'm going to talk to her about our clinical trial results. So we delved into that. I basically said, look, you've got to pay attention to this. Please, when you're done with this conference, go back and read our preprint. It's absolutely amazing. You're going to be astounded. The other thing that I kind of planted the seed in is I said, look, we need to work together because glycovirology, this is really the next big thing in science. We got to combine forces. People don't know who we are, don't understand how powerful this technology is. And here we're commercializing, things that you're researching, so, what better way for us to join forces? So, I planted a seed there and I said, hey, let's reach out. Let's discuss this in the future. So we'll see what comes to pass. But, I was pretty excited about that. Now, the other thing that I was excited about was the actual panel discussion that she was on. She talked about diversity in science. And I think that diversity is an essential element in furthering the progress of our science. So I thought it was really cool to see her speak about that. It was an all-woman panel, but still, diversity is diversity. And we need that to, expand where we're going. And our science is so unknown, it definitely needs more exposure. But now, the coolest part of the story wasn't the real meeting. The coolest part of the story was 3 days after we met. Because guess what happened? She tweeted back, and basically she called out. She called me out and said our clinical data about galectin inhibitors was, quote, exciting. When I saw that, I'm like, whoa, this is a scientist calling it something exciting. So we were really pleased with that. Now, investors don't get all crazy and thinking, oh, this is some sort of endorsement. But look, this is an acknowledgement that we really got something big. You got a Nobel laureate saying she's excited. That's big. Galectin science, we got 4,000 journal articles, and they all say the same thing. We believe a galectin antagonist is the best way to treat this disease. I mean, when you say that 4 times and don't listen to what the recommendation is, I call that a neglected science. There's only 3 active companies now, at least public companies, developing these type of galectin antagonists. So it's really nice and refreshing to at least have a Nobel laureate validate, through her understanding the potential of blocking galectins and what we're doing. That gets me really excited, and that's what was so refreshing about that interaction.
Awesome. Congratulations on that.
Yeah, thank you. Yeah.
You did mention galectin a lot. So for our investors who have never heard of galectin, tell us what they are and what they do.
Okay, it's really important because galectins are glycoproteins. So what does and where do they reside? They reside in the extracellular structure. So they're in between cells, and they recognize carbohydrates. Now, the most studied of galectins, there's a whole bunch of them. There's like 15 of them. But the most studied is galectin-3. And it has a snowflake shape that acts like an adhesion molecule. And it's responsible for a number of inflammatory types of responses. And a lot of them lead to tissue fibrosis. Now, snowflakes. I want you to think about snowflakes. What can you do? You can kind of pack them into a snowball and you can throw them at people. They're good adhesion molecules, aren't they? And what they do is they come together to form these voids between cells, and the way they come together is really called fibrosis. Now, let me talk about what. Okay, so now you have an idea what they are. So now let's talk about what happens when they kind of hang out inside your organs, right? They are a central cause of fibrosis. I want to put things in perspective. Fibrosis of the heart is kind of having like this really, really tiny mini heart attack. Fibrosis of the brain is like having a little mini stroke. You kind of get the picture. It's not good. So in almost all these cases, what we have is something called elevated serum of galectin-3. And or just galectin-3 in general is going to have a negative impact on your health. Now, like I said, there's 15 types of galectins, and not all galectins are bad, like, primarily galectin-3. But these other galectins help fine-tune the body's immune response. So they're immune modulators, and they keep everything in this nice balance and homeostasis. So they are important. So hopefully this gives investors a really good idea, an overview of what galectins are and how really important that they are.
Yes, that helps clarify it a lot. So talk a little bit about your clinical trials. What stage are you in there?
We'll start with our lead candidate. It's called Prolectin M, short, I call it PLM. Look, the end game of that molecule is to conduct a phase 3 trial with 408 patients with mild to moderate COVID. Now, in the meantime, right now is the meantime, we're conducting a dose optimization study, which is going to basically inform our phase 3. Now, look, we got all the regulatory approvals that we need to proceed. All we really need to do is basically pull the trigger and start it. We have another clinical trial that's also contingent on funding, like Prolectin I, for funding, like Prolectin I, all right? So Prolectin I, what is that? That's an intravenous galectin antagonist. We are going to start there with pulmonary fibrosis. Now, I'm excited about that because there's so many different uses of these an intravenous galectin-3. So once we get through our toxicity studies, and basically that's the phase 1, then it lends itself to rapid expansion, disease by disease indication. So we're really excited about that and the new opportunities.
So is Prolectin I just an intravenous version of Prolectin M?
A lot of people don't understand this, but the answer is no, it's not. They're two different molecules. Yes, these are two different distinct molecules with different approval pathways. Prolectin M, it's an oral tablet. It's designed to be chewed in the mouth. It kind of mixes with that saliva, making a paste, and basically coats this nasal pharyngeal area of your throat, mouth area, and it ends up being absorbed through the mucosal mucosa, say that fast, located in your mouth. That's why we believe it works so well, because it's really localized to this infected area when you get the disease. So now Prolectin I, this is obviously, it's I for IV, right? So it's designed for intravenous. Now, the nice thing about this, when we get it into the clinic, is we really don't have to worry about the bioavailability of going through the gut and making its way through the stomach acid. It goes right into the bloodstream, and it's like on target, on mission to basically quiet down that inflammatory process that ends up leading to fibrosis.
So what are the upcoming catalysts?
We got a number of catalysts. I think the value inflection point is our peer-reviewed journal article based off our latest preprint. I'm gonna break the news here. I think that's gonna be out in a couple weeks. We basically, we got the go ahead, and we're just waiting for them to, hit the submit button. So when that comes out, it's a little bit different than our preprint, but when that comes out, we believe that's going to be a value inflection point, because when you have such good data like what we have, the 100% responders rate, half the people, they're just not going to believe you. But then the professionals, the professional investors, look, their whole fundamental basis of a peer review, and all their investment strategies based on peer review. So here we have that. So now we have 50% of the audience that we at least believe is going to believe us now that we had a 100% response rate and take this seriously, including all the big pharma players. Because if you're a big pharma player, you want to base your career on a preprint or a peer-reviewed journal article, you see? So that's the first catalyst that I see. The next thing is we are, like I said, we're ready to hit the submit button. So expect the start of our dose escalation study. Another thing that we're expecting soon is we're expecting the clearance to proceed with prolactin I in pulmonary fibrosis. Now we should be getting that from the CDSCO. What does that stand for? Don't ask me, but it's the Indian FDA. We are also working on our clearance to proceed with our phase 2 study in the United States for mild to moderate COVID. So that's, I'm not going to say where that's at. I'm not going to announce whether we hit the button or didn't hit the button, but we're waiting for that clearance to proceed. We'll alert investors if and when we get the go ahead. And then the other thing that I'd mentioned is that we're looking testing on other viruses. For example, RSV, viral conjunctivitis. Those are some viruses of extreme interest. And then we're still working on journal articles, and we hope to see that for publication. So quite a few catalysts coming up here. We're in this super news cycle, I think.
Yeah, lots of good things on the horizon. So,
yes.
Mike, you mentioned before that there's a bunch of medical research that says galectins are implicated in a wide variety of diseases driven by galectins for each letter of the alphabet. So let's start at A for Alzheimer's,
yeah.
B for breast cancer, and looking forward past COVID infection, I'm assuming it's C for COVID. What would you say the next disease priority is?
I want to say something here. I think we connected the dots on long COVID, and I'll tell you why. There's 4 prevailing theories. Number 1, you got viral persistence. Number 2, tissue damage. Number 3, autoimmunity. And number 4, dysbiosis. Now, if all these theories kind of sound complicated, let's just talk about the top 2. Viral persistence. What does that mean? It means the virus is still hanging out in your body. Luckily, we have a molecule that addresses this. It's called prolactin M, right? The next theory is about tissue damage. Tissue damage comes from the inflammatory fibrotic process of COVID-19. Now, with our antifibrotic drug called prolactin I, we hope to reverse this process. Now, let's talk about the theory of autoimmunity. We can make this theoretical case that we can restore the immune system to normal function. That means we might be able to address this issue of autoantibodies. Then dysbiosis, look, that's a fancy word for saying, look, everything in the microbiome of the gut is just out of whack, and we need to put it back in order. And there's hints in medical journals that collect and control part of that process as well. So we're thinking we could have multiple shots at hitting long COVID, right? So I think that is probably our number 1 disease priority. But don't rule out all the other ones in the alphabet. So we're looking to have a crack at a number of other diseases. Now,
you also have a market cap of approximately 60 million. So how are you valued next to your peers? Is there some sort of formula that investors could point to in order to come up with a valuation?
You know, biotech's really tricky when it comes to valuation because no drug is the same. I mean, there's no like cookie cutter approach, but there is kind of a formula, and I'll talk about that. The formula that a lot of people use out there, that investors use, it's based on the stage of development. So that's why they care about. That's why investors, biotech investors, should care. Okay, what phase are you in? You know, because there's a number, there's a valuation typically assigned to that. Now, it's not just the phase, it also matters on other variables like the prevalence of the disease, the existing cost to treat the disease too, right? So if you have this really rare disease and it costs a lot of money, but it doesn't treat many people, you know, that's not worth as much as you know, something like cancer that costs a lot to treat, and there's a lot of people out there. So let's now take this concept of a formula and apply it to prolactin M, for example. We believe we're phase 3 ready stage. You know, that's how we think the market should view us. Okay. Yes, we're doing, you know, we're doing a dose optimization study prior to the phase 3 in India, but we believe we could call us phase 3 ready, right? So in this stage, the average drug, sit down, is worth 1.25 billion. And that's according to Baybridge Bio. Now he's basically a pharma consultant, but he did a lot of research on it. And you know, you can go to his website and you'll see all these numbers out there. Now, if we get really conservative and say, you know, this is a phase 2 asset, you're smoking something, right? Then we're talking about 285 million. Now, if we're talking a phase 1 asset, like prolactin I, because that's where we're at, well, then that's worth 100 million. So no matter how you slice this apple, we are undervalued, and grossly undervalued, given we have, well, we almost have 2 molecules. The almost 2 is the prolactin I. Now, these valuations are for just the average drug with the average side effect and just basically run-of-the-mill disease indications, right? That means that the Baybridge estimates are way, way too conservative. Prolactin N is a very highly efficacious drug with virtually no side effects. Nobody out there has anything that stops transmission the way this is proven to stop transmission. It stops COVID in its tracks in basically 3 days, 88% responders rate on day 3. We eliminated the viral load. I got to ask you investors, what is that worth? Is that worth the current $60 million market cap? If you answered no, I think it's worth more than that, then I agree with you. All right. Let's look at some other comps out there. So the other way to look at valuation are comps. Veru was valued at $2 billion at its peak. Now they had a COVID asset, and it, you know, and then they went to the FDA advisory committee. It had kind of a shaky response from the advisors. I don't know what they're going to do with it. You know, the market cap's down to $500 million, and maybe it gets something. But you know, the next one that comes to mind is another company called Atea. The stock symbol on that was AVIR. They were valued multiples of billions before they failed their phase 2, right? And then what about Pfizer's Paxlovid? Let's take a look at that. That's a comp, right? They're expected to do 22 billion in 2022, and Merck, they sold 5 billion already. So these drugs are usually valued at multiples of peak sales, and the peak sales is like 2 to 7x, right? So I got to look at this as a very highly undervalued asset of what we have. And then I'll throw one more out there. Pfizer bought Reviral. They bought them for their RSV asset for $525 million. That was a phase 2 asset. So think about, all we got to do is get to, possibly, I don't know if we can do anything with RSV. The news isn't out yet, but if we do something in that, look at, as soon as we get to phase 2, look at what it may be worth. So these comps are pretty amazing. And, I don't know what we're doing, but no matter how you cut the cookie or slice the apple, so to speak, I think we're undervalued. Now, why are we undervalued? Look, for the and I'll wrap this up. I don't know if how we're doing on time, but for the past 7 months, we had one investor that was selling over 60% of the stock daily. Luckily, we had a bunch of buyers. You, the investors out there that were picking up the shares, you kept us alive. So I want to say thank you. Okay. But now I want you to look at the volume. I want you to go to your screen. I want you to look at the volume. I want you to see how anemic it is and say, do I really think, and I want you to ask this question, do I really think that this guy is still selling? Once you figure the answer out, I think that we are going to go through a massive price discovery. What's price discovery? That's where your stock sees its true value. I believe in the market forces, and I believe that we're going to do that. You need volume to get price discovery. So hopefully we'll get to where I believe the price discovery will be at. And I hope investors take this message extremely well. And I don't know, Anna, I don't know if we have any more time, so I apologize for running over.
No, you're perfect. We are out of time, but I want to give you about a minute for any closing remarks, Mike.
No, guys, there's a real opportunity. I'm talking to the investors out there. There's a real opportunity to take on this pandemic. And the best way for us to do that is really getting this science and this technology to market. We can't do it without you. So I implore you, listen to the video, do your due diligence, look at this technology, look at the people backing it, take my Galectin challenge, and tell all your friends and family about it. People need to know about this. Thanks.
Yes, they do. Great job as always, Mike. Such a great presentation, and congratulations on all these milestones that you've hit. So we look forward to seeing you again in the near future with some updates.
Thank you.
All right. Thank you. Okay, everyone, stay with us. We'll be right back with our next presenter.